A nossa palestrante é uma pesquisadora britânica, ela trabalha com melhoramento de trigo. Então, por causa disso, eu vou pedir licença a todos para começar a falar em inglês, para que ela não fique deslocada também. E eu só peço que, se alguém tiver alguma dificuldade, não entender alguma coisa, sintam-se à vontade para mandar mensagem no chat. Pode ser no privado, pode ser para mim, pode ser para qualquer um dos integrantes do Gente também. Eu acho que talvez falar para eles darem um oi no chat para vocês saberem quem são. E, bom, é isso. Vou começar a falar em inglês. Welcome, everyone. Today's meeting is promoted by Gente. We invited Philippa Borrill. She's a plant science maiden working with breeding. I will present her briefly, but I just want some warnings before, not warnings, some reminders. We will have 45 minutes of a presentation, and after that we have more 10 to 15 minutes for questions. So if you can open your microphone, or you can send the questions in the chat. So we thank Philippa for accepting our invitation. Welcome, and thank you so much for sharing with us all this knowledge that we have in wheat breeding. That's a kind of a new thing for us here in the part in Brazil that we live. It's more common in the south, but now the Embrapa, that's a huge public institution here, is trying to stimulate the wheat fields in the center-west, Midwest, and I think that in the next years we will have intense work in wheat breeding. So, and also wheat is a polyploid crop and a model plant for polyploids. So I think your talk will be very important for us. So I will present Philippa briefly. Dr. Philippa is a lecturer in plant biology in the School of Biosciences at the University of Birmingham in the United Kingdom. She graduated from the University of Cambridge and obtained a PhD in biology from the John Innes Center. Due to her outstanding achievements as a PhD student, she won a fellowship also held at the John Innes Center to study the genetic networks controlling senescence and nutrient remobilization in wheat, and to investigate homology expression bias. During this time, she also worked on developing resources for wheat breeding, such as the Wheat Training website and the Gene Expression Atlas for wheat. The atlas was published in 2018, back-to-back with the International Wheat Genome Sequencing paper in Science. Currently, Dr. Philippa is a member of the coordinating committee of the International Wheat Genome Sequencing Consortium. Still in 2018, Dr. Philippa established her research group at the University of Birmingham. Her group uses a range of genetic and genomic approaches to understand how the process of senescence development determines the nutrient content of wheat grain and the regulation of the wheat polyploid genome. They aim to identify new targets for breeding wheat with higher nutritional value. So once again, thank you, Philippa, and feel free to share your screen and start.
Sure. Okay, so thank you very much for the invitation. I'm very happy to be here and share some of our research on polyploid wheat with you today. And thank you for the kind introduction. So my lab works on, as Letitia said, polyploid wheat improvement. And I'm sure that you're all familiar with the reason for why I'm interested in wheat. So wheat provides is an important staple crop around the world. It provides about 20% of calories eaten by humankind, and also about 20% of protein in the human diet. And like many other staple crops, we need to increase the production of these to meet the needs of a growing population, but also to manage in response to climate change. But to achieve this in wheat is particularly difficult due to its large and complex genome, which I'll come back to throughout the talk. So today I'd like to talk about ours and many others in the wheat community's work, where we've been trying to tackle this complex genome by building a suite of genomic resources which now enable us to understand in much greater detail the biology of wheat, and also opens up new opportunities for crop improvement. So I'd like to talk today a little bit about how wheat is a polyploid, why this is relevant, why we can now start tackling this challenge now, how can we make the most of polyploidy, and then how can we escape from polyploidy. So why is polyploidy important? So polyploid plants are very abundant throughout the natural world. So over 70% of flowering plants have been a polyploid at some point during their evolutionary history. And even more close to my heart, there are many of our major crops which are polyploid. I'm sure you recognize many of those on this slide, with my particular interest being wheat on the bottom right. So wheat evolved through several steps of hybridization, the first of which happened between two diploid ancestral species, Triticum iratu, the A genome progenitor, and the B genome progenitor, which is thought to be extinct but would have been similar to Aegilops baltoides. And these two diploids hybridized about 400,000 years ago to produce the tetraploid wild emmer. And this tetraploid wheat underwent a second hybridization event with another diploid about 10,000 years ago. And this hexaploid was domesticated into bread wheat, which is grown very widely in the world today. There's also an existing tetraploid wheat that's grown for making pasta, but that's grown on a much smaller area. So about 5% of wheat is the tetraploid grown globally, whereas about 95% is hexaploid wheat, which is the main focus in my research. So just to talk about the wheat genome in general terms, if we look at the genome sizes of several well-known organisms that many people study, so Arabidopsis, rice, and humans, these are their genome sizes represented proportionally. And then if we make wheat on the same scale, we can see the size of the wheat genome. So it's about 5 times larger than the human genome. And to make this more complicated, we also, due to this history of polyploidization, have 3 highly similar genomes, the A, the B, and the D. But I know in this audience, there are probably many people who work on even more complicated genomes. So this is sugarcane in the same scale. I only put 10 subgenomes. I know there can be up to 12, and these can be very variable between different cultivars. So in this circumstance, I feel very lucky to work on wheat, which has only 3 copies to worry about. So in wheat, we have 21 chromosomes in total. As you can see here, they belong to either the A subgenome, the B subgenome, or the D subgenome. And if we have a gene that's present on chromosome A, so here we have a gene on chromosome 7A, we will also very likely have a highly similar copy on 7B and another highly similar copy on 7D. And these 3 genes together are known as the triad, and I'll come back to this later. And these genes are homologs, and they tend to be very similar in their sequence. So around over 95% sequence similarity in their coding sequences. And this raises some questions about whether these 3 copies of most genes have a similar or a different function in wheat. And at the moment, we don't have a universal rule, we don't know, but I want to elaborate on this a bit in the next slides. So one of the reasons I'm interested in this is because I think this potential redundancy between homologs has lots of opportunities for breeding. If we were working in a diploid species, like rice, for example, if you mutate a gene that affects a specific phenotype, and you have a homozygous plant, you'd get a strong effect on phenotype. And in wheat, we can see this in some cases in genes which are dominant. So if you mutate just one of the 3 homologs, you get a strong effect. So this includes some genes that are well known to be involved in flowering, so photoperiod 2 and vernalization 1. But we also have a lot of genes in wheat that we know that have dosage effects. So you could have a case where if you mutate 1 copy, you get a 33% effect, 2 copies, you get a 66% effect, and all 3 copies, you get 100% effect of the mutation. And one such example is GW2, and this affects grain width. So what you can see here is wild type wheat plant, and then if you make a single mutation in one of the 3 homologs, a double mutation or a triple mutation, you can see a gradual increase in grain width, and also you get a gradual increase in 1,000 grain weight, showing this additive effect of the 3 homologs. Whereas in rice, the ortholog, where rice is a diploid, if you just knock out a single copy, you get a really strong phenotypic effect straight away. You don't have to go through this multiple step change. But I would just note, you do see a different overall change in rice. So it's 49% increase in 1,000 grain weight. Even in wheat, when we knock out all 3 copies, there's only a 21% effect, which might suggest these genes have slightly different functions between wheat and rice. As well as these additive effects, we can also see non-additive effects, where 1 copy is mutated, only a small effect happens. When you mutate 2 copies, you see a small but slightly bigger effect. But then when you knock out all 3 copies, you get a really strong effect on phenotype. And I'll show a couple of examples, and these include genes involved in grain protein content and also waxy involved in starch synthesis in the grain. And we have a third possible class as well, which are functionally redundant. So if you knock out 1 copy, you don't get any phenotypic effect. If you knock out 2 copies, we still don't see any phenotypic effect. And it's only when you knock out all 3 copies that you get a strong phenotypic effect. And some known examples of these include starch branching enzyme 2 and the MLO powdery mildew locus. And what's interesting about these different categories of dosage effects is that QTLs, so that we've been selected on in breeding programs, can only really target these dominant and additive genes where you can see a significant effect when only 1 homolog is mutated or altered in its gene sequence. So the consequence of this is that breeders have only been able to work with quite a limited amount of the variation that's available, and there's all this hidden variation hiding in these non-additive and functionally redundant genes, where mutating 1 homolog doesn't produce enough of a phenotypic effect to select for it in a breeding program. And we can see examples of this. So for example, GPC, which is a grain protein content gene that also affects senescence, we can see some quite strong effects. So if we have a difference between wild type and a version that's got 0 copies in the wild type, actually for this gene, then 1 copy is already mutated. Then you see a small effect in senescence. This is how this gene, and then when you mutate 2 copies, you see a stronger delay in senescence, so going to about 5 to 7 days. And when you mutate all 3 copies, you get a really significant effect in senescence, so of about 25 days. And just to put this into how it looks in the field, then you can see that at the top we have plants with the wild type, and then when you knock out all 3 copies at the bottom, you get this really dramatic delay in senescence. So if we were just selecting on the variation we could see, then we wouldn't be able to see this really strong phenotypic effect. But by knowing the gene, we can then knock out all 3 copies and have this really strong phenotypic effect. This is also seen for other genes involved in, for example, the starch biosynthesis pathway that have been known about biochemically for many years. So one such example is waxy, and if we have a wild type plant, it has 100% amylose in the grain. So amylose and amylopectin are 2 components of starch in the grain. And if you knock out 1 copy of waxy, you get a small but significant decrease in amylose content in the grain. If you knock out 2 copies, you get another small but significant decrease in amylose content. But if you knock out all 3 copies, you pretty much abolish amylose in the grain, showing that this gene has a really strong effect on amylose content. But if you just looked in a single or a double mutant, you wouldn't realize the full extent of the importance of this gene in this pathway. We also see an effect on amylose for the starch branching enzyme 2A. So in a wild type plant, if we set again amylose content to 100%, if you knock out 1 or 2 copies of this gene, you don't get any significant change in amylose content, suggesting this gene has functionally redundant homologs. When we knock out all 3 copies, we get a really massive increase in amylose content, showing that this gene really is important for amylose, but the 3 homologs are functionally redundant. So only when you knock out all 3 copies do you see a significant effect. So I think we need to understand polyploidy because it's so common in our major crop species. And also it has this serious consequence that it hides variation from breeders. So there's a lot of potential phenotypic variation that we have been unable to use so far. So for the case of wheat, why is now the time? Why can we start understanding this functional redundancy and tapping into this extra variation? So in 2018, we sequenced the wheat genome, and we were able to assemble over 94% of the wheat genome sequence. It's arranged into pseudomolecules, 21 of them, so each one corresponds to a chromosome. And we also annotated over 110,000 genes on the genome. And this is a vast improvement on the previous assemblies that we had that were much more fragmented. So now we have a much better understanding of the gene order, the whole order of the genome in its most of the genome, although some parts of highly repetitive sequence is still missing. But this reference sequence really provides a scaffold to start understanding many other aspects of genomics in wheat. So this is just a summary of some of the other resources that have been generated in recent years, which have been integrated together on this reference sequence from 2018. So having a reference sequence now makes gene editing much more accessible in wheat. We can start understanding DNA methylation. We can use targeted enrichment approaches like Rna-Seq to identify genes in specific gene families. And what I'll focus on more today is talking about RNA-seq data that we've been able to analyze using this genome as a reference, tilling mutants, which are chemically mutagenized lines, which are mutated in genes. And these are a really powerful resource to start unlocking these extended phenotypes. And also, finally, I'll talk about how we can use regulatory networks to unpick the effects of the 3 homologs. So first, using RNA-seq to understand global homolog expressions. So we collated together over 850 samples of RNA-seq data for wheat, and we're interested in looking at gene expression globally, for example, across a developmental time course. So here you can see some pictures of wheat at different developmental stages, and we harvested tissues from each of these stages and carried out RNA sequencing. So there are 70 different tissues and time points for which RNA sequencing is available. And we made all this data available through an EFP browser in collaboration with Nicholas Provart, Andy Sharp, and Yogendra Kedika. So it's a nice visual representation. If you have a gene of interest in wheat, you can go straight to this expression atlas and look where your gene of interest is expressed. We also integrated in another genome browser 1,000 RNA-seq samples, and this is an interactive genome atlas. So you can resort the samples according to their age. You can select a particular developmental stage that you're interested in. You can resort them according to the tissue that you want to look at. And you can also display the expression levels of the 3 homologs. So this is a really powerful resource with these different samples. So not just the developmental time course I mentioned earlier, but also from different cultivars, from different stress conditions, so drought conditions, heat conditions, and most of the samples we could find at the time that were publicly available were incorporated into this expression atlas, and we still continue updating it. If people publish a paper and they want to include it in this expression atlas, we're very happy to include extra data that's going to be useful to the community. And just to make a little advert, this expression atlas and the way the data is displayed is available to download, so you can put your own data. So if you add your own data, you can download a virtual machine that will run a local instance of this expression atlas. And there's 2 versions. There's one that already has pre-analyzed wheat data, so you could add more wheat data. But perhaps more interestingly, there's also one that's empty, so you can add your own transcriptome and then display your own data for whichever species you're interested in. And all the data is available on these web links, which I'm sure we can share afterwards if anyone's interested. So using all this data, one of our first questions was, we know there's many of the genes in the wheat genome have these 3 homologs. Perhaps the expression data can tell us something about their functional redundancy. For example, if one of the 3 copies is the only one that's expressed, maybe that's the important one that would affect the phenotype. Whereas if all 3 copies are expressed in the same way in the same tissue, perhaps they're functionally redundant. So we started to study homolog expression. And if we have a gene, for example, this gene NAC1 that has an A1 copy, a B1 copy, and a D1 copy, we can look at the expression level from the RNA-seq data, and we can measure expression level in transcripts per million. And this is a normalized expression level, regardless of how many sequencing reads you have. So we can compare the expression of the A genome, the B genome, and the D genome. So in this case, it's 10, 10, and 10. We can then normalize the expression patterns. So we decided to make sure that each of the each triad, which consists of these three homologs, A plus B plus D, should sum to 1. So in this case, then the A copy is expressed at 0.33, B at 0.33, and D at 0.33. And this means we can compare triads, whatever expression level they have. So for example, if we have a triad where the A is expressed at 80 and the B at 10 and the D at 10, we again can normalize it to be 0.8, 0.1, and 0.1. So this means that triads that have a very different total expression level can still be compared to each other. So we did this and first just looked at the global patterns. So across all triads, we found there were significant differences between the A, the B, and the D homolog in their expression levels. But a note of caution is that these differences are actually very small. So although they're highly significant because there are thousands of genes, the differences are 0.2% or 0.4%. So how relevant this is to the biology of wheat, we don't know. But we weren't just interested in global patterns, we were interested in what happens in individual triads. So you can plot the expression level of triads on a triangle graph. If anyone is an agronomist, you might be familiar with soil plots. So if we take a triad that has 30% of the expression from the A, 30% from the B, and 40% from the D, we can plot it on this triangle. So 30% A, 30% B, and 40% D, and then we can make a dot for the expression of this triad overall. So when you have a similar expression of the A, B, and D, the dot is in the middle of the triangle. If we have a different triad that has a different expression pattern, we can again plot it on this graph. So 21% A, 7% B, and 72% D, and we get a dot, which is much more close to the D corner because the D homolog is much more highly expressed than the other homologs. So what happens if we look across all the triads in the wheat genome? This is the overall pattern that we see. So we do see all the different categories of expression you could imagine. So about 72% of the triads have a balanced expression between the three homologs, where the A, the B, and the D are expressed at pretty much the same level. It's also fairly common to find genes that have one of the three homologs expressed. So 7% have the A homolog expressed at a lower level than the B and the D. 7% have the B suppressed, and 6% have the D homolog suppressed. And more rarely, we also see cases where within a triad, one of the three homologs is more highly expressed. So A dominant or B dominant or D dominant, about 2% of triads fit into each of these categories. So we were interested in how did this come about? Why are some triads balanced? Why are some triads suppressed or dominant? And how can we explain this through the evolution of wheat through these several steps of polyploidization? So what we did was we looked at gene expression data from a tetraploid and a diploid that can be then crossed together and resynthesized to make hexaploid wheat. And Rna-Seq data was, Rna-Seq samples were taken for these, the two parents, and also the synthetic hexaploid that had just been recreated. And we measured the expression in these different lines. So what we could do is take the expected expression of the tetraploid and the diploid. If you add them together, you can make a kind of pretend triad of what you expect you should see. And then we can compare that to what's actually measured in the synthetic hexaploid. And what we found was that mostly the balanced triads in the tetraploid and diploid that we would predict would have a similar expression of the A, B, and D, mostly remained balanced in the synthetic hexaploid. But for triads where you had, for example, A dominant or A suppressed, or B dominant or B suppressed, or D dominant or D suppressed in the tetraploid or diploid, when you added them together in silico, then we often saw changes when you went into the synthetic hexaploid. So in total, 47% of these non-balanced triads changed upon polyploidization into the synthetic hexaploid. We were also really interested in what happened during evolution. So wheat has been a hexaploid for about 10,000 years, and it was domesticated in this time. So we wanted to compare between the synthetic hexaploid, which is newly created, to an existing cultivar. In this case, we used Chinese spring, which is a landrace that's actually the same cultivar is sequenced for the genome sequence. And again, we observed in this time that most of the balanced triads remain balanced between the synthetic hexaploid and Chinese spring, whereas the non-balanced triads actually had an even larger change. So about 68% of them changed in their expression patterns between the synthetic hexaploid and Chinese spring, this current cultivar. So we observed significant transcriptional reprogramming, and this happens immediately upon polyploidization. So when you create the synthetic hexaploid, it also happens after polyploidization. So during this period of, well, we can't really say it's during the 10,000 years because we couldn't wait 10,000 years, but comparing this synthetic hexaploid to actually cultivars we see today. And these patterns are recreated in multiple pairwise comparisons. But it's also important to note that there were already some pre-existing differences between A, B, and D homolog expression level before the polyploidization step, and many of these differences were also retained in hexaploid wheat. So why am I working on wheat now? So the wheat genome sequences really formed a great platform to start analyzing biological questions in wheat, and we've been able to use RNA-Seq data to understand that many homologs in wheat are expressed at unbalanced levels. So while 70% are expressed at balanced levels, 30% have unbalanced expression levels. And perhaps that provides an opportunity for breeding if only one of your 3 homologs is highly expressed. Perhaps that's the one that should be targeted in breeding programs. So how can we make the most of polyploidy? I've talked about it as being maybe having some complications, but it does also have some advantages. So one great advantage is that this high level of redundancy between homologs allows you to have a really high mutation rate. So for example, if you use a chemical mutagen to induce mutations, you can add a very large amount of the chemical mutagen compared to an diploid species, which allows you to have a really high mutation rate. So to illustrate this, if you have a population size in different ploidy levels, so if we focus on the population size that's between about 1200 and 1500, the blue bar, if you have a diploid, you can only have about 380 kilobases between mutations. So you have to have your mutations quite far apart or else it becomes lethal to the organism. So if we have population size of 1200, then you only most likely get a truncation mutation in about 1/5 of your genes. But if it's a polyploid species, so tetraploid or hexaploid shown in the blue and red line, then you can add a lot more of the mutagen. So you can have a much higher mutation density, which means that actually even with quite a small population size, you have a really good probability to find a mutation in your particular gene of interest, which can be seen at the top of the slide, where with a population size of 1200, 1500, in hexaploid wheat, for example, 90% of genes will have a truncation mutation within them. So this is what we did. We took wheat seeds and added EMS with ethyl methanol sulfate, which is a chemical mutagen. We took the M1 mutant generation, selfed this to generate the M2 that would be homozygous for mutations. We extracted genomic DNA and collected the seeds to provide a stock for future experiments. And what we wanted to do was then sequence the genomic DNA to identify where the mutations were in the genome and make a catalog of mutants that people can order. But one of the challenges was that wheat has this really large genome and a lot of it is not coding sequence. So if we think about some model organisms like Saccharomyces cerevisiae, so yeast, a large proportion of the genome is coding and a very small part is non-coding, so repetitive sequences. As we move into genomes that are larger, things like Arabidopsis thaliana or Drosophila, then you have a much higher proportion of these repetitive sequence. And we were interested in identifying mutations just in the coding region to make it a bit more simple. So looking within the genic space. And this gets even more tricky if you're sequencing a human or wheat, because if you did a genome sequencing approach, whole genome sequencing, 98% of your sequencing would be focused on this non-coding repetitive region, which wouldn't have been useful in our case. So what we did was we used exome capture to focus on this 1 to 2% of coding region. And then we sequenced all of these different lines. So 1200 for the hexaploid wheat, and also an additional population of tetraploid wheat as well. So the mutations are all now available. So using this, we provided the data on ensemble plants. So if you have a favorite gene in wheat, you can go to your favorite gene and then you can look for which mutations are available through this population. So this is just an example. So each of the little boxes in yellow green or red, are showing what mutations are available. So you can pick out, for example, a stop codon mutation or a synonymous or a non-synonymous amino acid point mutation. On average, 90% of the genes have a stop codon mutation. So you have a really good chance of finding a knockout allele for your gene of interest. So this is great. We can now identify mutations in a gene of interest by just going to a website, which is really nice. But there's still another step to go, being a polyploid. So we need to combine the mutations together to make a double or a triple mutant. And we do this by crossing. So for example, in a tetraploid wheat, you would cross together a mutant in the A homolog and a separate line that will be mutated in the B homolog, self the F1, and then produce a segregating population that would make homozygous wild type for both the A and the B, and also homozygous mutant for the A and the B homolog. A similar process would need to be done in hexaploid, but we have the additional D homolog. So 2 separate crosses would need to be made, and it takes 1 more generation to get to a complete null mutant. And this is great, but it still takes quite a long time to cross together all these different mutants to get a complete null mutant. And as I talked about before, often if you just knock out 1 homolog, you're not going to see a phenotypic effect. So in general, we take the approach that we'll need to knock out all 3 homologs to get a phenotypic effect to really start studying this gene's function. But recently, a new technique called speed breeding has been developed by Lee Hickey and Amy Watson at Queensland University and Branda Wolf and Shreya Ghosh at the John Innes Centre. And this is really exciting to us because when you grow the plants under these speed breeding conditions, you dramatically accelerate their growth and their development. So here we have on the left a plant that's grown in speed breeding, and this is in effect 22-hour light with 2 hours of dark. So you have this really dramatic acceleration in growth. So you can see already these wheat plants have ears, spikes at the top, whereas in a normal growth condition, so 16 hours light on the right-hand side, the plant is much less developed. And what's exciting about this technique is that then you can move from it taking about 12 months to make a segregating tetraploid population, or maybe 18 months to get to a complete null mutant in hexaploid. You can roughly half the time it takes to make these complete knockout mutants. So this dramatically accelerates how quickly we can develop these mutants and characterize genes in wheat. So in wheat, being a polyploid, we can really take advantage of this fact and have high levels of EMS mutagenesis. And we've been able to create a tilling population which has a wide range of mutant alleles across most of the genes in the genome. And combining with speed breeding is really powerful to generate complete null mutants for phenotypic characterization. So how can we escape from polyploidy? So how do we know which genes we want to look at using some of these resources? So my lab is interested in senescence, which is a process that happens at the same time that the grain are filling. So gradually over the course of the growing season, the plants go from green through to brown, as you can see here. These are some aerial photos of plots in the field. And this process of senescence is important because the plant has a choice to make. It can either continue to photosynthesize, which could mean that it fixes more carbon, which could translate to an increase in yield, or it could start to senesce, which would allow nutrients to be remobilized from the vegetative tissues like the leaves into the developing grain, which could potentially improve the nutritional content of the grain and improve quality. So really senescence can be thought of as helping to generate a balance between the final yield and the quality or nutritional content of the wheat grain. But despite the importance of this process, and we know it's under genetic control, we didn't have much understanding of which genes were involved and how these genes were interacting with each other to regulate senescence. And because of the polyploid genome of wheat, if we tried to do a forward genetic screen for mutants, which could be normally done in a diploid species, we didn't really find any mutants with an accelerated or delayed senescence because of this problem with functional redundancy, because by chance in a mutant screen, you'll only knock out one of the 3 homologs. So we decided to use gene regulatory network modeling, which could tease apart the effects of the 3 homologs and therefore identify genes involved in regulating the senescence process that we couldn't be aware of before. So to apply this approach, we looked at gene expression across time using RNA-Seq. We then built a network and then use this network to identify the best candidate gene. So for example, in this example network, we have lots of genes, each gene is a circle, connected together, and the blue gene in the middle is very connected to many other genes, so that would make it a good candidate for regulating the process of senescence. We could then use the tilling population to identify mutants in our genes of interest, cross them together to make the complete null mutants, and then look at the phenotypes. So this is the overall procedure we followed. So we looked at a time course of senescence from 3 to 26 days after anthesis. And this is still quite early in the senescence process, so the leaves are still quite green. And the reason for this was that we were interested in the initial triggers of senescence, which will be the first genes in this process, because we thought these would be the ones we would most strongly affect the phenotype with if we knocked them out. And we harvested samples from these 10 time points after anthesis, and then did RNA sequencing on them in triplicate. And we thought about how we could analyze this data. One of our first thoughts was to use co-expression analysis, so kind of guilt by association approach, where if you have a gene which you know is involved in senescence, you can look at expression pattern and then find other genes which have a similar expression pattern and then infer they might also be involved in senescence. And we did try this approach. We found some interesting candidate genes, but we were much more interested in trying to identify genes that were completely unknown and wouldn't necessarily have the same expression pattern as a known gene. So to do this, we used a method called causal structure inference, where if you have a gene that has a specific gene expression pattern, so for example, this blue one goes up and then it goes down, we can then look at other genes and see how their expression pattern compares. So for example, this green gene, again, has a pattern that goes up and down, but this is slightly later in time than the original blue gene. And from this, we can infer that the blue gene is regulating the green gene. We might have another gene, this orange gene, that goes up, but its expression level stays high. So this orange gene wouldn't get included into the network. And we might have another gene that's earlier on in the time course, so it goes up and down, but before the original gene. So we infer that it comes even more upstream in the network. So by doing this, we can compare every gene to every other gene and work out if they fit together in this gene network and infer the regulatory interactions between them. But one of the problems with this approach is it's very computationally expensive because you have to compare each gene against every other gene. And the wheat genome is huge and has many thousands of genes. So this wasn't computationally viable. So we needed to narrow down our candidate gene list. So we went from the 110,000 genes in the wheat genome. We focused just on the genes which were expressed in our time course. This gave us 52,000 genes. We then narrowed this down further to only genes that are differentially expressed, so they change their expression level during the time course. Unfortunately, this was still too many genes, so we then selected only transcription factors. And this gave us 314 genes, which was a feasible number of genes to run the causal structure inference program with. And this is what we got out. I know it looks a bit of a mess, so I'm going to talk through it. So each gene is represented by a circle, and the lines in between them represent connections. And how big the circle is indicates how many other genes that the gene is connected to. So you can see at the bottom, there are some very big circles, which are highly connected genes in the network. And the other thing to note is this network has a directionality. So you should be kind of reading from the bottom to the top. So all the arrows on the lines, which I deleted because they were too small and messy, go up the screen. So the first thing we wanted to do was check any known genes that we know in senescence in wheat, can they be found in this network? And one gene that we already knew about was the NAM gene that has a dramatic effect on senescence. So we looked for this gene and its homologs in the network, and we found them all within our network near to the bottom, which are these kind of earlier steps which will be regulating many other genes. So that seems very promising. The reason there's 4 is because there's also a paralogous set of NAM genes in wheat. So this was great. It validated our network might be telling us sensible things. So the next step was that we wanted to then develop double mutants for candidate genes that were in the network. So as I mentioned, we decided to work on tetraploid wheat to save time. So we only have to knock out the A homolog and the B homolog to characterize these genes. We crossed together independent mutants to make an F1 and then self these to generate either wild types or double homozygous mutants for characterization. So which genes did we choose to develop these mutants? Well, the first gene we selected was this gene here, which is also a NAC transcription factor similar to the NAM genes. It's actually an additional copy of this NAM gene that we didn't know about before because it was missing from the genome assembly. So for this NAM2 copy, so a paralogous copy, we generated complete null mutants in this gene. And then when we look at these in the greenhouse, we see a really strong effect in senescence. So in the double homozygous mutant that's knocked out in the A copy and the B copy, we have a dramatic delay in senescence compared to wild type. And this is a quantification of that effect. We were also interested in identifying totally novel genes. So this NAM-A2 gene, you could say, well, it's quite related to the already known genes. Maybe it's not that interesting. So we then looked at other genes that were not closely related to the NAM genes. So this gene shown here in blue is another NAC transcription factor. There are about 350 NAC transcription factors in wheat, so it's not very closely related to the NAM genes. And again, in this gene, when we knocked out both the A and the B homolog, we saw a strong delay in senescence, as you can see on the left. So using this gene network approach, we've really been able to accelerate how quickly we can identify new genes regulating senescence. And we knew previously 2 transcription factors that regulate senescence, and we've been able to identify 2 new genes, and we're continuing to work to characterize additional genes from this network, including some of the ones at the bottom that I haven't talked about that are very large circles that seem like very promising candidate genes to look into. So in conclusion, then, we need to understand polyploidy because it's both a challenge but also an opportunity. And I think that in wheat, we now have many really great new resources that help us to understand the polyploid wheat genome. And I think that wheat could really start to become a model system for working in allopolyploids because of these great resources and answer some questions that are outstanding for many polyploid species. We found that homologs are expressed differently in wheat in many cases, which opens up new opportunities for crop breeding. And we've also been able to use gene networks to help to pick apart the effects of the different homologs and really start to understand individual gene function in wheat. In the last 2 minutes, I just want to say that making all these resources is great, but it's only useful if other people can use them. So we've also developed a wheat-training.com website that provides training materials for anyone who's interested in working on wheat. So if it is something that your institute might be moving into, I would highly recommend you have a look on this website. So we provide information about growing wheat in greenhouse conditions, but also maybe more importantly, all these different genomic resources, how you could apply them in your research, how you can access the resources, where you can order the seeds, all that kind of practical information. And this wheat training website has been a big community effort as well. In addition to the wheat training website, we have a wheat expression atlas that's free to access, and the EFP browser that I also mentioned. All the mutations that I've talked about are available on Ensemble Plants, and they've been really great partners to help host data and make it freely accessible. This kind of effort is really a collaborative effort across many different countries. So this is a paper we recently published that really evolved from the wheat training website. So we wanted to showcase all the different resources that are available for wheat. So imagining if you're a researcher working in a different species and you wanted to characterize your gene in wheat, how would you go about it? What would be the steps you'd want to undertake? So this paper really walks you through it. And it involved 27 authors from 12 different institutions across 3 continents. So it's really a global effort to bring these resources to the global community. And I'm also a great believer in training. So these websites and research papers are great, but sometimes you really need practical experience. So I've run several training workshops. So for example, through the UK Cereals Network Monogram and also a UK plant science network called Garnet, we run hands-on training sessions to learn how to use different bioinformatics tools and access all these genomic resources. And I'd just like to end by thanking everyone in my lab at the University of Birmingham. I would also like to thank Chris Bawawi at the John Innes Centre and members of his lab. A lot of the work I presented was carried out whilst I was still at the John Innes Centre. I'd like to thank the International Wheat Genome Sequencing Consortium and many, many authors who helped us in the Rna-Seq studies that I've talked about. And I'd like to thank all the funders of this research, and I'll be happy to take any questions. Thank you.
Thank you, Philippa, for your talk. That was amazing, learning a bit more about wheat. like, at least for me. If anyone has a question, you can put it in the chat, or you can open your microphone if you feel comfortable.
So I have a question.
Okay.
So Dr. Borrill, I was thinking about the knockouts that you were doing on the plants to knock out the genes. Were you using your Tillian population, or were you using another approach?
Yeah, so they were using the Tilling population. So we just look for our gene of interest, identify, we focus on stop codon mutations so that cause a premature truncation mutation. We generally try to choose the earliest mutations possible in the gene model to make sure we get a full knockout. If you choose one quite to the 3' end of the coding sequence, you might not, you might just remove a small proportion of the coding region. That might not have such a strong phenotypic effect. So that would be our preferred option. In some of these lines, we did actually have to rely on missense mutations because sometimes you're unlucky and you don't get a stop codon mutation because it's random which mutations you can find in each gene. But luckily, this NAC transcription factor family is very well characterized. So from model species information, we could predict which amino acids would be detrimental if they were mutated. So yeah.
Do you think, I'm sorry, do you think is there any room in your lab to use like gene editing approaches, like if you can multiplex, like to target many genes at once with your guide RNAs? Do you think is that possible? Let's say 30 genes at a time.
30 genes at a time. Okay, I think that's quite a tall order. So one of the limitations of doing gene editing in wheat is that wheat is still not that easy to transform. So you have to do like embryo rescue and tissue culture. So it is getting a lot better. So there's a recent publication that came out about a GRF GIF overexpression line that can really improve transformation efficiency. So some previously untransformable cultivars can now be transformed, but it is quite a bottleneck and it's quite labor intensive and quite expensive. So yeah, to make a transgenic line in wheat maybe costs $3,000 or $4,000 US. So it's quite an expensive procedure. In terms of technical feasibility, lots of people really want to do CRISPR so they can knock out, say, the A, the B, and the D homolog simultaneously using the same guide RNA, which I think is kind of what you're alluding to a bit. And a few people have been able to, but you need to have so many transformants to get the edit in all 3 homologs that it's not that feasible unless you're a lab who does the transformation in-house, which my lab doesn't do at the moment. But yeah, I think it would theoretically be possible to edit multiple genes at once, but I think the transformation is still a bit of a bottleneck in wheat. And also in Europe, then GM crops are not very acceptable in terms of farmers wanting to grow them or the public as well. So for my research, I like to use tilling lines because they're not regulated as GM organisms, so you can readily apply them in the field. And breeding companies would be interested to work with you. If you're working with CRISPR and transgenics, then they're not so interested in your, well, they're interested in your results, but it's not going to be immediately applied in their breeding programs. So the wheat in the UK, then lots of research is done in the public sector in universities and research institutes, whereas most of the cultivars that are released are all released by private breeding companies. So to get your work into the field, you need to partner with private companies who obviously are interested in commercial returns. So, yeah.
Thank you.
We have a question here in the chat.
Yep.
It's Professor Gabriel. He says, thank you very much for the great presentation, Dr. Borrill. My connection is really unstable, so he will ask the question here in the chat. With 95% CDS similarity, how difficult is to reliably assign RNA-seq reads to homologous? Is there a lot of variation among genes?
Yeah, so it's a good question. It was also something we were quite worried about too, or like took account of when we wanted to analyze the different homolog expression, because if we couldn't clearly assign reads to one of the three homologs, we couldn't really say that the A genome is more highly expressed than the B and the D. So we did all our mapping using Callisto, that's a pseudo-aligner, and it seems to be really accurate between the three homologs, even though they have this 95 to 97% identity. Callisto has it uses a probabilistic model to assign the reads to the different three homologs. So although you might have some parts of the gene which are identical between the A and the B and the D, it calculates based on the adjacent reads and then assigns the reads proportionately according to the regions it could distinguish. So that's helps with telling the difference between the three homologs. And also in wheat, we're lucky to have lots of cytogenetic stocks. So we actually have lines where, for example, the whole of chromosome 1A is completely deleted. And instead you have 2 copies of chromosome 1B, which then the wheat plants can grow completely normally. They're called nullotetrasomic lines. So we use some Rna-Seq data from those lines to check if we had accurate mapping of the homolog. So if you delete the whole of chromosome 1A, it's not there at all. You can tell by looking down the microscope. You shouldn't see any expression from genes on chromosome 1A. And that's exactly what we saw. And for the chromosome that's completely doubled, then we saw twice as much expression level from genes on that chromosome. So we are quite confident about this homolog specific mapping. And I think that the level of, I guess, dissimilarity is sufficient to be able to tell the different homologs apart. But I know in some other polyploid species, people use other methods where they only keep reads that have SNPs between the three homologs to be able to be sure about the mapping. But I think because wheat's sufficiently different and we have these cytogenetic stocks, we can validate our pipeline is accurately distinguishing homolog expression.
Great, thank you. Any more questions? We have time for more, 1 or 2 questions.
I have more questions. Since you came all the way from England to Brazil, so let's use your time. Did you think about that heading wheat to see the effects on senescence? Like a sudden effect, maybe you can identify some genes?
We haven't done that. I think there are quite a lot of physiological studies that have been done before trying to manipulate source sink ratios where they, for example, take away 50% of the developing grains and see what that effect that has on the senescence. So in general, when you reduce the sink strength, so you get rid of some grains or you cut the whole ear, then you would get a delay in senescence. But to my knowledge, no one's looked at a kind of gene expression level, what's going on and how that would relate to the senescence we've been looking at in a more, I guess, natural situation. But yeah, it'll be interesting to see how the networks and responses of the plant would adapt. But I suppose there'll also be quite a lot of stress responses happening if you dehead the wheat plant. Yeah.
More questions? I have one question. Actually, I have many, but and how do you deal with the plant cycle? So senescence is variable from very different cultivars, and when you are evaluating different cultivars in the field, for example, trying to find an interesting gene or understanding how a mutant is going to behave. How do you deal with this different time of maturation?
Yeah, so it's difficult. So with all the data I presented, then we normalize to the heading date. So then we have, even if the plants flower 1 week later than the other ones, then for example, we count senescence from when did heading occur in that particular line. So it's days after heading. So that normalizes for kind of developmental differences that would happen before. In terms of comparing different cultivars in the field, that's really difficult because they have so many different phenotypic differences. So it's hard to tell, is the difference you're seeing really related to maybe genetic variation in the gene that you're interested in, or is it due to other background effects between those different cultivars? So in a lot of cases to validate the genes, we're now relying on these mutant populations where we can specifically knock out one gene, do some back crossing to clean up any background mutations, and then we can compare sibling lines that will have pretty much the identical genetic background, just differing at one particular gene of interest. So that's adjusts for that issue as well. And then also when you phenotype senescence in the greenhouse or you phenotype it in the field, it's really different. In the field, you're at the perils of the weather and what happens in the UK. It's not very predictable. So every year is different. So if you're doing a field trial, you need to run it for about 3 years to get some kind of reliable results. So it's quite challenging.
We have time for one question. Okay, I have one other question, but it's more related about the genomic resources in the database. Yeah. Here in Brazil, we are facing a kind of lack of investment in science. So I believe that from now, many students will need to try to find other way to get data than producing their own data. Do you think these resources can help these students so they can find data in this database and develop an entire studio with that? For example, downloading entire Rna-Seq data and try to understand another mechanism, and which database would be addressed to this situation?
Yeah, so I think it's a really great question. It's something I'm really interested in, is data reuse, because a lot of people generate a lot of data, they analyze it for one particular question they have in mind, and then the data is kind of going to waste. So there's a lot of other questions you could ask with that data. So the expression atlas that we built that now has over 1,000 RNA-seq samples in it, then I think we put about 300, but the other 700 plus come from publicly available RNA-seq data that we just downloaded. So we tended to use the NCBI short read archive, but also the European Nucleotide Archive. I think they have like the same data. They're freely accessible. You can download all the FASTQ files from people's RNA-seq experiments. One thing that can be quite difficult, so in 2016, when we first started setting up this expression atlas, I was the person who was lucky to go through the whole database and see all the different wheat samples, try and work out, someone's uploaded this RNA-seq sample, what experiment did it come from? What cultivar is it from? What condition was the plant grown? Which tissue is it? All this information is not very well recorded in these databases. So it takes quite a lot of manual curation to work out what the sample is, and some detective work to try and match it to the paper to work it out if it's the same sample or it's a different sample. So that's one kind of, I guess, just to highlight a potential issue that you could come across, is trying to identify exactly what material you have in these databases. But I think there's a lot of potential in that. So for example, I didn't talk about it today, but we used a lot of these samples to generate tissue-specific co-expression networks to try and identify genes involved in specific tissue developmental processes. And this was nearly all based on publicly available data. So if you have a question you want to answer, I'm not sure for sugarcane how much data is out there, but for many species, there are a lot of RNA-seq samples that are out there. So I haven't looked recently, but in wheat, about 6 months ago, there were more than 2,000 RNA-seq samples that you can just download. So there's a huge quantity of data. It's working out what's your question. And then I think there's a lot of opportunities that are out there. And in wheat, because genomics has moved so quickly, we've gone through about 5 genome sequences since 2014. So every time you get a new genome sequence, there's new opportunities to reanalyze old data that perhaps was analyzed using an incomplete and older version of the genome. And now with a new improved genome sequence, you can actually find out new findings from the same data, even with the same kind of biological question, if you like, because previously some genes might have been missing. So you miss some interesting biologically relevant answers.
Nice, thank you. If anyone else has a question, it's already 5. So I would thank you, Philippa, again for the really nice talk and for being available at evening here to talk to us in a very crowded week. actually. That is, she's running a big event that I was talking in the beginning. So thank you again, and welcome everybody one more time, and thank you for attending.
Well, thank you very much for the invitation, and thanks all for coming. So yeah, have a nice evening.